I decided to opt for an umbrella today rather than a hat because it was so wet. I thought, this is rain. It's Yep, yep. Well, it would be messy and not that much fun in town. But I mean, it'll be lovely skiing at Happy Jack. Yeah, the snowies right now are fantastic for cross skiing when it gets warm during the day. You know, if it gets warm during the day. Well, if it doesn't heat up, then you don't get a crust on the top layer. Yeah, which is what we like to crust cruise. You know, you can skate anywhere. Yeah, it's cool. It is probably one of the coolest things in the world. Yeah, I mean, it's basically like floating across the landscape. Like, oh, it's the best. It really is.
So it's second to the 11th.
Yeah, and occasionally I get it done sooner, but I mean, I never count on that. So, but that gives you a pretty large window of time.
When you give our poster presentations, does it switch or like what do you do?
Yeah, let's see. Poster presentations are on the regularly scheduled final exam day, which I don't remember. I think that's Wednesday. So if the 11th is Wednesday, then yeah. I think it's on the Wednesday. Yeah. I've received a few logistical questions, and some of them have come to me via email and some of them in person this morning, but I want to try to answer them as well as I can. I believe that the one that was emailed to me regarding your unknown report that you're all working on for lab was actually just a formatting issue. So for the introduction for your unknown report, that is the section in which you will cover background information. Tell us about why it's very important to be able to identify bacterial unknowns, both in medical field, but also in other disciplines, food microbiology, environmental microbiology. There are a lot of areas in which we want to be able to find out what is a particular isolate that we've taken from some environmental niche or some human niche, if you will. And that'll go into your intro. You're also going to discuss how we do that, like how we go about identifying an unknown. And soon in class, we will actually talk about genotypic strategies for that. You're all using phenotypic strategies right now because you're actually looking for visible characteristics and capacities, metabolic, physiological traits that you can actually somehow detect. We'll talk a little more about how you can also look at DNA sequences to help you identify an unknown. But making mention of that, perhaps in your introduction, is fine, that you can either identify genotypically or phenotypically, and that you've been focusing on the latter. So that's the meat of your introduction. One thing about an intro always, as just a general writing rule of thumb, is that an introduction should begin broad, like a funnel. It should funnel down to more and more and more specific until finally you state your hypothesis, which is the most specific part of your introduction. And then your results your methods are all about how you tested that hypothesis, and your results are all about what you found, allowing you to either accept or reject that hypothesis. So it's pretty cool. I almost think of it as an hourglass, really general up here, coming down to your specific hypothesis, addressing that specific hypothesis, and then at the very end you get really broad again and say how this might contribute to the larger big picture. So you kind of come back around and come back to why it was important to be able to identify that unknown, what larger ramification could it have, and so that's how the overarching report will look. But please, I hope that helps to characterize or to help you with the introduction is not separate from the background information. That was a question I received. The background information is a part of your introduction, but so too are your objectives and hypotheses. There's a formatting issue, and that was what was confusing you in the assignment description. Does that help?
Yeah.
Okay.
Thank you.
Good. Now, the other question I received regards the final exam. The final exam is fully take home. It's open book, open note, open resource, open me. You can use absolutely everything. But that also means that it is a very difficult, critical thought-based assessment. It means that it will be very time consuming. Depending upon the person, it may take 12 hours or more to finish the exam. So you do need to get going on it right away. I do hand it out on the 2nd, I believe it was the date that we looked up. And then it's tentatively due on the 11th, so you get a pretty large window to work on it. But be diligent during that window in working on it. And do bear in mind that it does include, that lab material is open there too, so it really is going to ask you to synthesize connections between everything that we've learned and put it together. It's going to be fun, because you'll think, oh my goodness, my mind has gotten more sexy than I ever thought it could be. So making those connections, you'll celebrate it, but you'll also have to work hard at it. So it maybe is not even too soon to put chunks of time into your calendar where you've set aside time to work on that exam. It's a great idea, and I'm glad that Katie brought it up today. Okay, any other logistical things that just happened, Jade? Yeah, so that's a great question, because what you're noticing is that the 2nd happens before the end of class. So yes, and it'll be fun because you can bring your final exam to class, and as we cover, as we cover a topic, you can, talk with one another and say, oh, I think the answer, she just covered it, look at that, woo. And you have that done, yeah, until then. Yes, roughly. The one big difference is there is no opt-out option. So all of the questions, you'll answer them all, because I figure you're not in a high-pressure environment. So otherwise, the format's really quite similar, and I think that works well. But of course, I am always up for discussion there. If you think it would be better to just get rid of the multiple choice, questions and just have free response, or, by all means, hit me with suggestions if things have occurred to you as you've been experiencing that format and there's something that you think we should change. Okay. You like it? Yeah. I mean, it works pretty well. Yeah.
It tests your understanding to where there's 2 different ways to grade the test.
Mm-hmm. Right.
Upon your strength.
Yes, it does allow that weighting, and that's what Riley is saying. So how I weight it, where either I weight your multiple choice more heavily or I weight your free response more heavily, and of course the final would be like that as well. So whichever section you performed the best on would be weighted the most heavily, which is nice to bring out the strengths of individual students. Beautiful. Anything more besides that on this beautiful snowy day? Hopefully you're all enjoying getting a little bit of moisture. And maybe it's making you think, gosh, this would be a great weekend to stay inside, maybe build a fire, and work on an abstract, right? I was thinking that myself, so I'll be reviewing abstracts. I got myself somehow into this responsibility of, for Phi Beta Kappa, we're giving 2 $500 awards for undergraduate research day for students who do the best integrated research, where they integrate liberal arts and the sciences in their research. And 270 students said, I think I qualify for this award, so now I get to review all of those abstracts. So I'll be reviewing abstracts by the fire while you're getting yours done and submitting that to me, or maybe working on homework 8. I didn't put a due date on there because I reckon there's a good chance that it will get elongated, but please do think about working on that. And of course, if you would like to come and review the 3rd exam, I would love to do that today. And for tutors, if you wanted to pick up a copy of the exam, I know some of you had expressed interest in that. Today would be a good day to do that as well, if that just happens to be an open time for you. If not, please see me at some other time and we'll get that taken care of. Okie doke, I think that covers logistics for today. And as we remind ourselves where we were last time, I've drawn a cheesy picture on the board. Just felt like a little cheesy board work today. And what method of lateral gene transfer seems like might happen in this picture? Transformation, because notice that this is naked DNA. I was going to add the racing flats on it, but that is naked DNA, free in the environment, Maybe there was another bacterial cell here that lysed and suddenly, released some of this naked DNA. Maybe a lot of it got sort of chewed up by nucleases, and so maybe there's these little fragments of linear DNA that are just free in the environment. So there are a couple things, please.
Are those naked strands single-stranded?
So what am I showing in this case?
Yeah.
Double-stranded DNA, yep, but
Circular. So
linear, linear double-stranded. Ah, you are like literally 2 steps ahead of me. Like you must be a good chess player, because seriously. No, we're going to come back to Riley, because what his question is could this linear double-stranded piece of DNA get integrated into this chromosome? And that's about 2 steps ahead of me, so we're going to come back there. But right now, I just want to tell you a couple of basic rules for all 3 methods of horizontal gene transfer, whether we're talking conjugation or transformation or transduction. All 3 of them follow these 3 basic rules. So just bear in mind that it's unidirectional. Number 1. Now, this seems sort of sad, because it seems like if you're undergoing conjugation and one bacterium hooks up with another bacterium and sends across some DNA, it seems only right that the bacterium that receives it should be able to say, "Oh, dude, thank you. Let me send you back some of my DNA." It doesn't work that way. Nope. This is totally unidirectional. So it's just donor to recipient. In eukaryotic systems, we see types of conjugation where it's bidirectional. Not in bacteria. It doesn't happen. So it's always just from donor to recipient. Okay? Now, hang on. If the transfer is chromosomal, meaning that maybe that naked DNA right there came from the chromosome of a lysed cell, only ever is a small piece of the chromosome transferred from donor to recipient. So if we think that through in the world of conjugation, what we can say is that if 2 bacteria are connected by a sex pelus, never is there going to be a situation where the donor bacterium says, "Hey, I'd just like to stay hooked up for a real long time, and I'm going to transfer my entire chromosome to you." Not a thing, okay? It's not going to happen. Only ever is a small piece of the chromosome transferred, no matter which type you're talking about. And then finally, this last one, I thought I could kind of pick on some of your groups in lab, because this is an experiment that you actually did. So, I think Coulter and Connor, you guys are hanging in Carrie, you guys are hanging out in lab, and you get really engaged in what you're talking about, and you totally forget to label the plates. But you know that you had one plate that was a negative control. Remember that one? That was the LB plate all by itself. And onto that LB plate, what did you put on it for a control? It may be a trick question. What happened on that LB plate that was the negative control? There was no antibiotic in it, so maybe we should say the opposite of nothing happened, which is everything. Yeah. There was no selective pressure on that plate, so an entire lawn of bacteria grew because there wasn't any antibiotic. Now, another plate that you had was the plate with the antibiotic. Now, only what kind of cells could grow on that antibiotic? Resistant cells. What kind of sexy term could we give them to describe them in lingo? They were? Fit. Well, yeah, this is true. They were definitely fit for survival on that plate. But what would we call them? Because what did they get? They got the DNA, right? They got the plasmid. The naked DNA that they took up was the plasmid that allowed them to express antibiotic resistance. So what would we call them? Transformants. Or we could also call them recombinants. So on that plate, only the successful recombinants or transformants grew. So even though Carrie and Coulter and Connor forgot to label their plates, how would, at the end of the day, Carrie could go back on there and he could say, I know that which one of those was the one with a successful recombinance. Left or right? Left. Which one was the negative control? Right. Okay? And so the conclusion that you just came to is point number 3. Recombination never happens in 100% of the potential recipients. That is not ever does every possible recipient of that naked DNA take up that naked DNA and start expressing the genes on it. In fact, at best, transformation efficiencies are around 10 to the minus third. How would we say that in layman's terms? 1 in every 1,000, at best. But often it's more like 1 in every million. It's common to see that kind of transformation efficiency. Or how would we write that? 10 to the minus 6th. Good.
How is that communicated to most cells, or is it just through happenstance chemical processes that it happens at?
Good. Good. So the answer to that is that it varies. And I think this is Riley's question. He's asking how is that communicated between cells? And I think what he's asking is how does a cell know there's naked DNA in its environment and it should take it up? Or likewise, how does a donor cell in conjugation know to connect to a recipient cell? How does that work out? And there's more than one answer to that question. There are some cells that have special competence factors on them that recognize naked DNA in their environment and that literally are membrane-spanning proteins called competence factors that say, hey, bring in that naked DNA. So it depends on the cell. Some of them recognize it really readily and others are not going to. And we're going to come back to that as well. Beautiful job. Now, okay, let's think our way through something else. Here's our naked DNA in the environment. It could get taken up by this recipient cell. So I want you to think about it. Now, Riley, you can answer that question. What is one possible fate of this naked DNA? And let me quickly just tell you that only one of the 2 strands are going to get in. So we'll only see one of these 2 that actually get taken up by the cell. What is one possible fate of it? Beautiful. This is where am I allowed, since it's Friday, to be R-rated a little bit? Okay, if you don't like it, close your ears. So there's this fantastic stand-up comic routine that Ellen DeGeneres does where she's talking about people that videotape their sex, and that she's like, well, how does that work? Is that kind of like a coaching thing? So, you know, you go back to it a little bit later and you kind of review it and give each other advice, and you're like, no, okay, that's really good. That was good. Oh, that, not again. Don't do that again. But like, and how about right here? It's like, energy, nothing, right? And this is what I think about fate number 1, if you will, because this is like energy, nothing, because there's this whole horizontal gene transfer event that takes place, right? And then boom, and we would say that fate is degradation. So it's literally, we could say it in a technical way, horizontal gene transfer can occur without resulting recombination. So energy, nothing. Nothing actually happens because it really just took up that DNA and then it degraded it. What's another possible fate, though, of that pink strand? Maybe it won't get degraded. Good, wonderful. So the other possible fate is that maybe it will find a region on the recipient cell's chromosome. By the way, what's the technical term for that chromosome in this case? Endogenate. Good. And then this was the exogenate. Good. So it's possible that there's a region on the endogenate that has striking similarity sequence-wise to the exogenate. And so therefore, this little pink fragment of DNA, and I probably showed it relatively too large there, right? It finds a region that it has similarity to, and it hydrogen bonds to this region that it looks like in the endogenate. And then, boom, it gets cleaved in. And this is what's really weird. The endogenate actually, so there's actually this pushing out of the existing DNA, it's like it creates this little bubble where it kind of inserts itself in place of the original DNA, and then this gets cut off and gets degraded, so it's what gets degraded, and then the pink DNA literally, oh, there we go, takes the place of that original orange DNA that's now been degraded. So that's stable incorporation, and we would now say that this recipient cell is yes, transformant or recombinant. Excellent job, yeah.
So then are you going to have a ton of missed base pairs?
Right. Beautiful. So the question then is this, we know it bore striking resemblance to this strand, but it's not identical. So are you going to have a bunch of messed up base pairs? Potentially. And the question that I pose to you is, what are the chances that with mismatch repair, a cell that is the recipient is going to fix those? What are the chances that it will fix those correctly? Maybe, okay, let's say it this way.
What they're paired with, because it's more likely going to fix two, like, purines or two.
Okay, yeah, no, I like that. So first we have, what are the chances of finding the mismatch? They're higher because we're not necessarily dealing with tautomerization now, and it also maybe does depend on what the mismatches are. So I like Riley's point that if you have a purine-purine pair, then it's more likely to be found by mismatch repair. Good. So, but the problem, or I guess the question I'm asking is, say it is overt, and let's say that it is very likely. Okay, so Hannah said, yeah, it's pretty likely, they're going to find it. But the problem is, what are the chances of fixing it correctly? Right? How does it know? Right? Yeah. They're both maturely methylated, right? Meaning that they both bear the kind of signs of mature DNA. So the chances of correctly fixing it are what, mathematically? 50-50. 50-50, yeah. So here we have an interesting way in which we see genetic diversity take place, right? And the way in which we might see changes that might allow for perpetual natural selection in a population. So that's, yeah, it's really cool. Hunter.
With this new gene, or the new DNA coming in, is there a problem with the promoter binding and the single factor binding, especially if that new piece that came in was similar?
Yeah.
To
Yeah.
other pieces, and so therefore it was just like totally screwed up everything?
Maybe, right. So could one foresee a situation in which a recombinant might die due to having recombined? Yes, it could happen. And one could also foresee a situation in which it could get a selective advantage. So there's a chance of either being the ramification. Yeah, I love it, Patrick.
This doesn't just happen spontaneously, right? When there's enzymes carrying the naked DNA and inserting it.
Actually, no. That's super interesting. There are enzymes involved, right? What enzyme do we know has to be involved with that whole process? Like, okay, yeah. So the enzymes that maybe, like ligase, that maybe knit together the ends, and then also what had to be involved with getting rid of that? Endonuclease, right? And then other nucleases that might chew down. And so yes, that, yeah. But what's interesting is that the cell is naturally, and I guess maybe I should say there still are enzymes involved with this, but cells are actually naturally inclined to allowing one piece of DNA to replace another. And one of the reasons for that is because of SOS repair. So this is SOS, right? It means just like, whoa, help, right? It means when there's a lot of damage to a chromosome, they actually make strands that can allow, replace really highly damaged strands. And so this is something that they're naturally inclined towards doing, as opposed to this like weird anomaly that would never happen in a cell. So yeah, but yeah, there still are enzymes involved in that. Let's write this down before we forget where we were. So if it contains a sequence that is homologous to that of the endogenate, One of the fates may be that it integrates, and that's the fate that we just finished drawing. We also know that we could have the degradation fate, and that's actually the one that I've listed fourth. But there's some interesting other fates that we didn't come up with. For example, that little pink piece of DNA might for some reason persist in the cell, but not get degraded, but not integrate. And in which case it would be present usually for one generation, where it might encode for some products, but it might not stay for future generations. There are some situations where it does perpetuate, which is really interesting, where it persists and replicates outside of the endogenous. And this is something that happens more in transduction than in transformation, and I'll show you that in a moment. We talked about it surviving but not replicating, and then of course our final fate of degradation. So there's some really interesting things that can be the fate of the exogenous.
Is it possible as well for like that extra piece of DNA to be incorporated into the plasmid?
Oh, that's a great question. So what if that DNA bore striking resemblance to a region in a plasmid? That could occur in a plasmid. Yeah, that's an interesting situation to envision. Maybe a little less likely just based upon likelihood of sequence similarity, but it's a good, yeah, it could happen, yeah. Okay, let's look at the details of transformation. You all remember this picture from lab, so it's not new to you. But one of the things that I have maybe capitalized on a little bit more or tried to show a little bit more is this possible fate of degradation. So uptake of naked DNA, and then I've gone ahead and added into this the possibility that that DNA will simply be degraded and there will be no productive recombination that took place as a result. Why, who has an idea why on the right, where the DNA being taken up is a plasmid? Remember that plasmid could either be a naturally occurring plasmid in the environment or it could be man-made. And that is something important to think about, is that most plasmids that we use in the lab are actually man-made for our purposes, to get bacteria to express things we want them to express. But does anyone understand why I might not have shown the degradation fate as, as much with the plasmid, Scott?
Nucleuses are kind of like Pac-Man, the outside edge.
Yep.
So it's circular.
Yeah, which has such interesting discussion questions to it. And I loved, so who's in the gaming group this semester? It's so cool because your leaders got to present at the Center for Teaching and Learning last week, or no, this week, Wednesday. And they did an excellent job. But it's interesting to think about the one game that you play where there's plasmids in it. And the truth is that is actually not as fictionalized as you would think, because what has been found is that human cells are naturally competent, which means that they take up DNA, we will take up naked DNA. And in fact, if you augment that just a little bit, using something that we call electroporation, a tiny bit of an electrical shock, we can really effectively deliver DNA to human cells. So think about what are some of the interesting applications of that. While we talk about the fact that a lot of those same terms are used in the bacterial world. So let's look at this linear, because you followed this in lab, right? But for now, I want to follow this for our purposes of lecture and look at that. So cells that can be transformed are termed competent. And so like I said, human cells are naturally competent. There's a lot of cells in the bacterial world that are also naturally competent. And I had just mentioned that, right, Hans is feeling rather incompetent today. And so he's not the happy cell taking up the naked DNA. But okay, so, which I absolutely adore this cartoon, but what would we critique about it if we were going to critique anything in it? What is true? I mean, I prefer that most of the cells are happy and competent, but what would be more likely? Yeah, that the minority, right? That would be switched to, really the minority in the population would be taking up the naked DNA. But it is worth saying that some populations are much more inclined to this than others are. So Pseudomonas, for example, ironically loves to take up naked DNA. And so we recognize it being one amongst many that are naturally competent. Azotobacter, Agrobacterium, Bacillus, Streptococcus, these have natural competence factors that they produce. You can see why a lot of soil bacteria would have evolved mechanisms for taking up naked DNA in order to perpetuate their chances of survival in a highly competitive soil environment, right? They like to see, okay, can we take up something that might give us a selective advantage? And they're in these environments teeming with cell lysis and possibility for taking up that naked DNA. So Streptococcus has natural competence factors. So does Haemophilus. Haemophilus has actually it takes in DNA with a little vesicle, which is unusual in bacteria. So depends on the bacterium how they express competence. Yeah.
Does this mean that all these naturally competent bacteria are more likely to be resistant to more things?
Okay, yeah, so that's a great question. So within the population of, say, Azotobacter that tends to be naturally competent, are we more likely to see that population being resistant and thriving and surviving in a highly competitive environment? Sure, it gives them potentially selective advantage. Yeah, good. Good. So lots of bacteria that are naturally competent, but some have to be made that way. And maybe this is like our deep philosophical discussion for the day is, you know, why is it that in the lab we constantly insist upon forcing bacteria to do things when there's whole populations of other bacteria that would do it naturally, but we're like, no, we want to use E. coli because it's easy. And so we use E. coli, and then we have to force it to take up DNA. We have to heat shock it. Or maybe it's a population that really isn't naturally competent, and then you have to electroporate it, which means, yeah, no, quite literally, you put it, and I was trying, Jeremy took this picture because he worked with an electroporator in his lab, but you can see that you put this cuvette that has metal sides on it, and you put it in between two electrodes, and you send a current through it, and you try to get it to shock the cells enough that they take up the naked DNA. And as I said, this is something that can be used in human cells as well to get human cells to take up DNA. The really funny thing is when you accidentally get too many electrolytes in the solution and it arcs and you get sparks all over the lab. That's the fun day. But that's one method that you can use to get them to take it up. Did anyone think about the question that I posed about why we might want to make human cells take up naked DNA? What might be an application of that? Yes. So vaccination, DNA vaccines. You know, if you can introduce a DNA sequence that encodes for an antigen that allows the body to recognize that as foreign invader and to mount, you know, a small immune response, then of course you will be vaccinated. And a DNA vaccine can be maybe more effective than other types of vaccines. So this is definitely an active area of research. And in fact, it's one I haven't actually looked at the literature body for a while. So if anybody's got, Delta, you had your phone out, if you want to just look up really quick and see where we are at like the forefront of gene therapy that uses DNA vaccines. But let's follow the fate of this linear strand of DNA. There's terms given to all of these processes, so whereby the single-stranded piece of DNA enters into the cell. I guess it doesn't take us much of a challenge to say that this step is called entry, as the single-stranded piece of DNA is being brought into the cell through competence factors potentially. So this one strand enters, but what I didn't show on my cartoon is that the other strand is degraded as the one strand enters. So it's like there's a co-nuclease function that occurs at that competence factor. So one strand gets in, and we know that it could get degraded. We know that there's some other fates, but I want to follow the productive integration fate, the one in which we see it getting replaced. And so in a lot of ways, this is just terms for what we drew on the board. So once inside the cell, we see that it positions by H bonding like we talked about, and we say that is homology, similarity. So if there's a homology between the exogenate and the endogenate, that's when we see it aligning. And then we call that step where it breaks the original endogenous DNA and cleaves it, and allows then through reunion the exogenous DNA to replace the endogenous. So this is just giving terms for what we drew already on the board. So breakage, the cleaving, the degradation of the original, what was our orange strand on the board, and then reunion was when our pink strand came in and hydrogen bound and replaced the original orange strand. Here.
Is the purple strand that was degraded by
The second strand of the red DNA. Yeah. Oh, yes, right, good. So in our cartoon, the purple was the one that would have been degraded upon entry. Beautiful. Yeah.
Single strand of DNA isn't exactly a small molecule. Does it have a carrier, or how does this happen?
Right. So the competence factors that we're talking about are actually quite well evolved transmembrane, integral membrane complexes that are very adequate for bringing in the DNA. But let's face it, when we're talking about forcing bacteria to take up naked DNA that don't normally have those competence factors, we actually don't quite understand.
You wouldn't really denature their membranes completely?
Well, like, do you...
Like electroporation?
Right. No, it's not enough to kill them, but it is enough to cause something, and we don't fully understand that something, because when they're naturally competent, we understand that they have these protein complexes through which the single strand of DNA goes. But when they're not naturally competent, we don't 100% understand why shocking them allows the DNA to get in. Yeah, Hannah.
I was just curious about why the exonuclease only degrades one strand, what keeps it from just degrading all of it?
This is a wonderful question because it is so super applied to what we know. What is true of double-stranded DNA? If we took the two ends and they were aligned against a cell, what would be true of one end and not the other? 5', 3'. And most exonucleases are 3' to 5' exonucleases. They chow on the 3' hydroxyl, and they don't on the 5' phosphate. So there's a real elegant distinguishing between those two ends. Yeah, good question. Yeah.
So the uptake of the DNA could technically be kind of random because
Right.
one end is 3' and the other end is the opposite.
Yep.
Flip it around,
Yep.
they switch the
You got it. Yep.
So
Yep. Beautiful point. Yeah.
So you say they're homologous and they're
Yep.
complementary, but they're only sort of partially complementary.
Yep,
Otherwise
correct.
it wouldn't do anything.
Good. And please do bear in mind that homology doesn't mean identity, right? They're not identical. They're just very, very similar. So again, that does mean that there could potentially be stable changes that result from recombination. And there are, right? There definitely are. Okay. Good. This is the same cartoon as we have on the board where you can see the exogenous sort of wheedling apart the endogenous and sticking itself in the place of the original DNA. Then, of course, that gets chowed by an endonuclease, meaning that it cleaves internally at the sites where the endogenous has been replaced. Those, of course, then are degraded. The recipient DNA, that region is degraded. And then, as you all told me, DNA ligase is needed to seal the nicks. Okay? And we talked about the 50-50 chance that a change will be fixed correctly. So yes, there will probably be stable changes from this. Okay, transformation. We are so ready to move on to transduction, right? Everybody feel good about looking at viruses? Okay, because that means you're going to be on the stage because I want you to rack your brains for just a bit, and I want you to think about what were the two very different kinds of phages that we discussed in lab. So that is your challenge at this point. are the two different types of phages that we discussed in lab. Yeah.
Go to do this electroporation on that machine.
Yep.
Do you just control the current?
The current.
Because it seems like
Yep.
direct current and electricity is just a derivative of radio waves. And I know that when you hit certain stuff with the right frequency of radio waves, they actually their pores spread apart.
Yeah.
Because they're basically just hyper
Yeah.
resonant.
Yep.
And so it seems I wonder with bacteria if you're just resonating to where those layers just get so spread apart.
Yeah.
And if you hit them anymore,
Yep.
they would shatter.
Yeah. Yeah.
But you're just resonating into
Yeah.
the point that they're
Yeah.
just about to break.
Yeah, yeah, and I think that'll be worth relating to Patrick too. But yeah, I think that is we do know like that in some way there is certainly a widening, enough of a widening that it gets in. Yeah, yeah.
If you ever watch water get hit with the right wavelength, it actually starts to cavitate and look like it's boiling, but it's not.
Uh-huh.
And like the surface of it would
Yeah.
expand because the distance between the molecules is amazing.
Yep.
And I
Yep.
bet that's what's
Yep. Yep. Who has two? I know this tends to be one of those questions where you're like, I think I remember a distinguished type. Who has just one type of bacteriophage? Catherine, go ahead. Of bacteriophage. Okay, good. Virulent. Okay, good. And what was the other one? Temperate. Nice. Yeah, good. 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 So we could go ahead and say virulent and temperate. And did anybody like draw out what the difference is between what they do at all? Yeah. So which one lyses? So we have virulent and temperate. Which one lyses? Virulent. Good. And then why might we say that temperate is like really the smooth criminal? Yeah, so it gets in, right, and replicates itself, but it doesn't really do anything to the host cell. It's like it's such a smooth criminal. It just gets in, it integrates itself into the genome of the host cell, and then it just hangs out. And it gets replicated with every binary fission event. So every time the host cell replicates itself, the viral DNA gets replicated as well. So they're getting propagated every single time the host cell divides. So what causes them to think, maybe I should get the heck out of Dodge and lyse this host cell? What might cause stress? Yeah, like when the host cell is in trouble, then the bacteriophage says, this is not such a good time to be just chilling out here. I'm going to go ahead and excise from the genome and get the heck out of Dodge. I'm going to lyse this cell and go on to another host cell. Wow. Because what does that, so that type of phage, the temperate, right, it keeps its host alive. And if you're a virus, that makes a lot of sense. What kind of viruses in humans are the smooth criminals that are like that? HIV. Good example. Yeah, right. Keeps the host alive, right? That's pretty smart. If you're, you can't anthropomorphize viruses, but I mean, right? It does make sense if you want to propagate and continue to replicate to keep your host alive. Okay, good. So let's talk about then, based upon these two types of bacteriophage, we need to talk about two types of transduction, right? Because in the lytic pathway, when the host cell is going to get lysed, and the bacteriophage are making many, many hundreds of themselves, they're making all their capsids, all their tail fibers, their base plates, their tail pins, and they're packaging DNA, phage DNA up into those capsids. Sometimes they make a mistake. Right? Instead of packaging phage DNA into, say, one of the many hundred capsids, they accidentally package DNA from the bacterium. Maybe piece of a plasmid, right? So what limits the amount of DNA that a phage can carry? Size. They're tiny. tiny, right? So there's like a max of 1 to 2.5% of the genome of a host cell that they might be able to fit into that tiny little package. But yeah, they could potentially take any DNA. One thing though, why might it probably, it could be, but probably wouldn't necessarily be a full-on plasmid? What does the phage do first to the host cell when it's like literally hijacking it? One of the things it does first. Yeah, it degrades the host DNA. So there's a ton of exonucleases. So like think of it, this is like a battleground where there's just all of these parts of host DNA and phage DNA all, around, but the host DNA is typically lysed. So it's typically little pieces of it that would get put up into the phage capsid. Yeah.
Is there any way, like chemical signaling within the host cell that tells itself that it's being attacked? Like, is there a
Yes.
mechanism
Yes.
directly against that?
Yes. So there's a kind of a separate conversation. What Riley asked is, does the host cell have any way of defending itself from being hijacked?
Because we've always talked about
Yeah, what would be one thing that it would do to try to defend itself from that hijack?
just lysine.
Yeah, recognizing foreign DNA, which we're going to talk a lot more about viruses later, and so we'll come back to that. But sometimes it tries to degrade, but sometimes it can't recognize it because viruses are smooth. They will try to look as much like the host as they can look, right? So Chantel, let me write a few things down, and then I'll take your questions. So remember that what we're describing here is the process by which a phage becomes defective. That is, if you're a phage, you want to package your own DNA into your capsid, not the host cell DNA. And when it's defective, it means it accidentally packaged host cell DNA into that capsid. So then it will be transferring host genes from one bacterium to the next. It won't be productively, if you're a phage, you wouldn't think of it as productively infecting another cell. Instead, you're like a taxi cab carrying DNA from a donor to a recipient. So 2 types of transduction can occur. One is generalized, and this is the one that we've been talking about. So generalized literally means when there's this battleground chaos from hijacking, the accidental putting of host DNA into the capsid instead of phage DNA, this is generalized transduction. It occurs during the lytic pathway of virulent bacteriophage or during the lytic pathway of temperate bacteriophage if they enter into that pathway, right? So this is kind of like a parts is parts, wrong thing happens. Now there's a second type, specialized transduction, and this only occurs in temperate bacteriophage because remember they're the smooth criminals that integrate into the host chromosome. And so sometimes there's a situation where, say the going gets rough for the host cell, and this integrated DNA of the phage, it starts thinking, whoa, it is time to excise and get the heck out of here, go through the lytic pathway, and leave this cell that's in duress, right? There's a lot of signals of duress, get out of there. But occasionally it screws up. It literally excises wrong. So when it excises, instead of just taking its own DNA out, it accidentally excises offset so that it takes some of its own DNA and some of the host DNA. And so in specialized transduction, it's only the genes that flank the site of integration of that phage. Does that make sense? That's what's specialized about it. It can only take these certain genes. I'm going to show you a picture here in a moment. So only a few types of specific genes versus in generalized, any gene could accidentally get packaged into that capsid. If it's specialized, it's only those that flank the site of integration. Now here's what's really interesting. That site of integration is uniform. So temperate bacteriophage recognize a specific sequence in the bacterial DNA where they tend to integrate. So they tend to be flanked on either side by two genes that reoccur in all cases. The bio gene is one of those. So I'll show you a picture of that. Does that make sense? Basically, what's generalized versus what's specialized. And this is next a picture of specialized, where you're seeing here, this is a picture from Miami University. Here is this smooth criminal. It's going to just go ahead and get in next between the gal gene and the bio gene, and it's going to integrate. And as long as the going is good in the host cell, it just stays. You know, things are pretty good. It gets replicated upon every cycle of binary fission. Every progeny cell has a copy of the phage. So it's propagating. But if things go wrong, then it tries to excise. Well, here we're showing that that excision, if normal, takes just phage DNA, the pink. But if it's got a problem, it could excise wrong. And now notice how it takes the flanking gal gene with it. So that means that this is now going to make a defective phage, and one that's going to carry that gal gene to the recipient cell, and it's a false threat, right? The phage is no longer effective, but it does act as what we call a generalized transducing particle, meaning that it can allow transduction to take place. Okay, Chantel, thank you for waiting on me to get that through.
I have a couple questions.
Okay.
So one of them, the flu virus, how exactly does that work in us? Like, I'm assuming we're talking
Mhm.
bacterial with this.
Yeah, good.
So how does the flu virus,
Yep.
like, does it do the licensing of our cells?
Yep.
How does that work?
You got it. So the question is, how are human viruses similar or different from bacteriophage, and specifically the influenza virus? The influenza virus, sadly, is not a very smooth criminal for it, right? Happily for us, it means that it gets into our lungs, which are the cells that it finds its receptors on, and we'll talk more about how that works later. It finds its receptors on our lung cells, it gets in, it hijacks them, it lyses them. Our immune system is like, whoa, foreign invader, there's cell death happening all over the place here, get in there, fix the problem, and within about 7 days, your immune system has gone ahead and cleared that infection, unless, you know, there's complications of that, right? And so it is an acute lytic kind of cycle that the influenza virus uses versus HIV, right, which is more along the lines of the smooth criminal where it integrates and it's in your genome, kind of like the temperate bacteriophage, only it randomly integrates, not, you know, different kinds of sites in our cells. Beautiful questions, and I can't wait until we actually are going to soon be talking about human viruses. So have a wonderful weekend.